जी। Yes, ma'am. Yes, ma'am. Audio. Hello. Hello. Hello, ma'am.
Yeah, ma'am. Okay.
Okay.
All right.
Volume bound, ma'am.
Volume.
Okay, okay. Unmute.
Thank you,
Send us
ma'am.
the
Thank you.
link.
Thank you, ma'am.
Dear participants, a very good morning. On behalf of the principal of St. Joseph's College for Women, Autonomous, Visakhapatnam, Dr. Sister Shaiji, the management and faculty and students of St. Joseph's College, we welcome you to this national webinar in biochemistry. St. Joseph's College, a 62-year-old college in North Coastal Andhra Pradesh, and Visakhapatnam, a very fast-moving city, is one of a very premier institution, and we stand for so many qualities which our motto is quality and excellence, hallmark of St. Joseph's, besides punctuality in starting our programs on time. On behalf of the college, once again, on the Department of Biochemistry, I, Ms. Ramani Dhanraj, Head of the Department of Office Management and member IQAC, welcome you to this one-day national webinar organized by the Department of Biochemistry and the IQAC on trends in biomedical instrumentation. This is the 12th webinar of the college in the corona pandemic season, and many other departments' webinars are underway. Bioinstrumentation or biomedical instrumentation, a field filled with biomedical engineers, as the punchline is, they are the hidden heroes of COVID-19 crisis. Biomedical engineers design biomedical equipment and devices to aid the recovery or improve the health of individuals, ranging from PPEs to ventilators and so many other important devices, by using their ingenuity to minimize creating time and optimize quality. And we all know, with the hugely increased demand for ventilators and other equipment over the last few months after the pandemic broke, we've seen the important role that medical technology plays in patient care. Today's speaker is a very dynamic researcher come academician from the prestigious Banaras Hindu University, Professor Saripella Sri Krishna, who is going to speak on a very relevant area in the COVID pandemic. And we know the COVID pandemic has resulted in a lot of disruption around, and we need the blessings of God to keep us sane and harmonious. Let us start the webinar with the prayer song, Lord, Make Us Instruments of Peace.
Lord, make us instruments of your peace. Where there is hatred, let your love increase. Lord, make us instruments of your peace. Walls of pride and prejudice shall cease when we are your instruments of peace.
This is what we need, friends, every day. Lots of peace as the number of cases are on the rise. I now invite Dr. P. Mary Anupama, Assistant Professor in the Department of Biochemistry and the coordinator of this national webinar, to highlight the rationale for the webinar. Over to Dr. Mary Anupama.
A very good morning, everyone. As the coordinator of the webinar, I, Dr. P. Mary Anupama, from the Department of Biochemistry, St. Joseph's College for Women, Visakhapatnam, extend a very warm welcome to the resource speaker, faculty, participants to this one-day webinar on trends in biomedical instrumentation. Biomedical instrumentation is an ever-growing field that deals with developing devices and instruments for measuring physiological parameters. During this COVID pandemic, since the past 3 months, we have been listening to terms like testing kits, ventilators, and all instrumental support systems that sometimes causes fear in our minds. The manufacture of few indigenous devices like fast detecting RT-PCR kits to Agua, the mini toaster-sized ventilators have been scaled up to help us meet the increasing demand. Our esteemed resource speaker, Professor Sri Krishna from the Department of Biochemistry, Banaras Hindu University, will deal with a very vibrant topic, correlative light and cryo-electron microscopy, technological revolution and biomedical applications in his session. I now invite Dr. Mausmi Shankar, Assistant Professor, Department of Biochemistry, to introduce our speaker, Professor S. Sri Krishna, to the audience. Over to Dr. Mausmi Shankar.
Ma'am, start.
Thank you, ma'am. A very good morning to all the participants. I'm Dr. Bausmi Shankar Adala, Assistant Professor of Biochemistry in the Department and St. Joseph's College of Women. It's indeed my greatest pleasure to introduce an eminent academician, Professor Saripalli Sri Krishna, Professor in Biochemistry, Banaras Hindu University, who has extraordinary research-based knowledge in the field of biochemistry, molecular biology, to name a few specialized fields. Having completed his graduation and post-graduation from Andhra University, he did his PhD degree from the very prestigious Banaras Hindu University, Uttar Pradesh, and continued his teaching career in the same university. A visiting scientist at Stanford University, California, he is an adjunct professor of Interdisciplinary School of Life Sciences of BHU. He pioneered several prestigious projects sanctioned by Government of India and also UK. Currently, he is handling a coveted of 5 crore rupees project of DBT COVID Screening Center at ISLS BHU. He has more than 40 national and international publications to his credit. He is an esteemed member in more than 11 national committees like Institution of Eminence, Advisory Committee of Imaging Facility, Central Discovery Center of BHU, Board of Studies, Aligarh University, Chairman of Smart India Hackathon, Treasurer, President of MHRD Innovation Cell, Deputy Professor in charge of Institute of Sciences, etc., to just name a few. Professor Sri Krishna mentored 4 PhD students and 5 more were pursuing their research besides 45 project thesis guiding. Not just attending many national and international conferences, he organized the 26th National Congress of Parasitology in the Department of Biochemistry of BHU, in addition to conducting several personality development programs such as UGC-sponsored Special Summer School, advanced courses on genes and behavior workshop, refresher and orientation courses. His resource talks are innumerable to diverse audience, and those who attended his session have always been enriched in ways inexplicable. With this truly inspiring profile, I cordially request Professor S. Sri Krishna to share his thoughts on this topic. Over to Professor S. Sri Krishna.
Thank you, Moshmi, for very nice introduction. Could you please let me know whether you are able to hear me or not?
Yes, sir, we are able to hear you.
Thank you. First of all, let me thank Dr. Rupani ma'am and Dr. Anupama for giving me this opportunity to interact with wide variety of students. I believe they are from heterogeneous population. ranging from college to PhD students across India, maybe across the globe. Considering this heterogeneous population of students, also considering the theme of the webinar, I try to explain things as simple as possible to reach the larger audience in very, very broad terms. Coming to my topic of the today's talk, I'll be talking on correlative light and cryo-electron microscopy uh, in terms of technological revolution and biomedical applications. The topic I selected because of two reasons. One is because of the current pandemic that is going on around us, and we should also think everyone, whether it is educationalist or maybe a general public, to think and orient themselves to address these kind of pandemics in future. Right now, the pandemic that we are facing is extremely severe and different from rest of the pandemics that we have experienced before. This everybody is witnessing. So considering this, I designed this talk so that it can reach to larger audience. As title indicates, it's a correlative light and cryo-electron microscopy.
So are you sharing your this thing, sir? PPT, sir?
Yeah, I'm sharing.
Please,
Is it
sir.
visible?
It's not yet seen, sir. Just share
Now,
screen.
is it visible?
Not yet, sir.
Not yet.
Yes.
Is it visible now?
Yes, sir. Please put it in slideshow mode, sir. Click
Yeah,
on
sure.
that. Click and it'll go on that, sir.
Yeah. So the title, as indicated here, is about correlative
Thank you, sir.
light. Welcome, ma'am. Light and cryo-electron microscopy. Uh, Everybody knows about microscopy. Whoever is in line of education, they must have gone through, or maybe they must have even witnessed what is microscope. So light microscope, everybody has seen, but the latest one is the cryo-electron microscope, which is extremely useful in understanding subatomic level of structures, which is extremely important to address various biomedical issues. Right now, the pandemic made us uh, on our toes to identify the structure of this tiny virus, which is causing extreme difficulties to general public. So in this figure, the extreme left figure shows the light microscopy image taken by scanning electron microscope. And the middle figure shows transmission electron micrograph of a single virus particle. You could see the spikes around the surface of this particle. And same thing is depicted in schematic in the right side of the picture. Inside the core protein, I think you can see a thread-like structures, which are nothing but the RNA, that is the genome of the virus. So you can just look at the size differences between the extreme left and the extreme right. The small yellow dot seen in left picture is one such virus particle. You can see millions of virus particles attacking the cell, and cell is in dying mode. So I will be talking about the following points during my 30 to 40 minutes talk. And these outlines are like evolution of biomedical instrumentation with respective to image per se. I'm not going to talk about ventilators and other instrumentation because that is not covered in this title. Then I'll be touching upon evolution of microscopes, early versus current trends, what latest we are using to address these issues. Then I'll be touching upon resolving structures of cells, molecules by both light microscopy as well as cryo-electron microscopy. The ET indicates the electron tomography, which is the latest available in very, very few labs across the globe. Then combining these two, can be called as a correlative microscopy, which is very, very latest in its budding stage. And this is very, very effective to understand the biology of the disease, whether it is virally induced or bacterial induced or both together. So that is where I'll be concentrating on my talk to give you more idea. So we all know that medical field extensively uses various instrumentation like X-rays. It has revolutionized the diagnosis of the medical field. Likewise, ultrasound, echo, angiography. We all knew this in day-to-day basis. Whenever we visit hospital, doctor routinely prescribes these different methods through which they can diagnose the patient and they can assess the condition of the patient. Accordingly, they can go for treatment. MRI is another interesting one, which has made a significant contribution towards the medical diagnosis. But what you may not have heard is fMRI, which is known as functional magnetic resonance imaging, which is largely used to understand the functionality of the tissues or organs of the body, okay, which is very, very fine in tuning the functional aspects of the organisms. So especially this has been worked out extensively on brain disorders. Like you can even understand the differences between the functionalities of the speechless versus speech, vision versus visionless individual brains. So that is what is there in the medical industry and medical field. People are actually using these very efficiently, extensively in the diagnosis front. Once the diagnosis is done, proper diagnosis is done, then what next is the treatment or maybe a therapeutic intervention. So proper diagnosis is always important for both the treatment and therapeutic interventions. As my talk is on imaging of the microscopy related aspects, I would like to bring to you about the size. Size is very, very important parameter of the imaging. Imaging is all about resolution, and we know that variety of microscopes are being used to resolve different structures. Depending on the size of the structure, depending on what aspect you would like to study in that within that structure area, you can choose the kind of microscopy that you would like to use. And this particular picture shows three different arrow marks. In first case, it is unaided eye, which can resolve anything from 1 millimeter and above, as big as you can see. But below 1 millimeter, you need some aid in terms of instrumentation. And that aid is nothing but microscopy here. And from 1 millimeter down to 1 micrometer, you have a range of microscopes which can be used to understand structures and functions of the molecules, which are very, very nano in structure. Below micron, you need to go for electron microscope. Most of you must have heard about transmission electron microscope as well as scanning electron microscopes. These two are used routinely to understand one surface details of the structure that you are looking for. That is what you call scanning electron microscope. I'm not going to focus on that today. The other one is known as transmission electron microscope, which can be used to understand structures as tiny as ribosomes, which are roughly about 25 to 30 nanometers in size, and proteins, and then lipid bilayers, small molecules. Very recently, last week, I think we have got a Nature publication where a group of scientists have demonstrated even atoms. They could resolve single atom from platinum. So therefore, I'll be focusing more on this light as well as electron microscope in today's talk. If you look into the evolution of the microscopes right from the beginning, like you have bright field microscopes, which routinely used in day-to-day experimentation in many biological science labs, and also maybe in the hospitals also people use for routine cell counting and other experiments. There is another microscope called dark field microscope, where the background is dark and you can see bright structures, so that you can, without staining any with any dye, you can observe the organism under dark field microscope. But the problem with this microscope is you cannot resolve to greater extent. The resolution limit is low. Third class of microscope is phase contrast microscope, which is used to understand phase differences which are existing within the cells. Because as you all know, being a biologist, cell has different organelles and different structures concentrated in particular areas. So when incident light is passed through the cell, wherever there are thick structures, there I think light will be retracted a bit, and there you can see a dark structure. Wherever there is no such thick tissue available, light can transmit freely. There you can see a bright field. So that bright and dark contrast can be assessed through phase lenses. Therefore, this microscope is better than the earlier two, which is routinely used to understand differences in phases. And the other three kinds of light microscopes, which are colored here in fluorescence, confocal, and super resolution. These are really very, very high-end microscopes of the light microscopy category, and which are not routinely used in any medical field. Mostly they are confined to research laboratories. I would suggest medical personnel should also adopt these microscopes in order to diagnose better, or in order to understand the pathology, disease pathology in a better way. Of course, the cost price also would increase from bright field to super resolution to the tune of say 10 crores. A super resolution microscope would cost around 5 to 10 crores depending on the features, specifications. Then next one is cryo-electron microscope. I'm not going to talk about transmission here. I'm talking about only cryo-electron microscope, because which is very latest. This has been made public after, I think, Nobel Prize winning research done by 3 scientists. I'll come to that little later. If you look into the early world of microscopes, you have microscope used way back in 15th century. This was the first microscope used in 1590, and then everybody knew about Robert Hooke, who has made available that cell hexagonal structure in 1635, somewhere in between 1635 and 1703. And he has used this microscope, which is very simple one, having objective lens and then ocular lens. Louis Pasteur in 1860 has used a little advanced microscope with the condenser lens, which is further improved by Carl Zeiss from Germany. He has actually put this condenser and other features to it so that the light path can be concentrated and focused onto the specimen of interest to view in greater detail. But all these microscopes were very primitive and they don't have much resolution. But in those days, they were the best microscopes. In current microscopy world, you come across microscopes like live imaging system, which is based on fluorescence imaging, and also you come across confocal microscope, which are in different forms. They can be multi-photon confocal and simple confocal microscope, or super resolution confocal microscopes. So you can look at the complexity of the systems. As you go further, microscopes are evolved much more complex, and cryo-electron microscope is much more complex. I will show you towards the end of this talk. Sure, we can see cells with good microscopes available till date, but then we cannot really identify their identities of the proteins or enzymes or any complex structures existing within the cells. You can see cell, but you cannot differentiate between proteins, because most of the cells or their structures, subcellular structures, or colorless. Then how can you visualize them? So we can color them to visualize. For example, in the left side, the cell which is harboring this green, red, blue, different colors, which are indicating specific molecule of choice or your interest, which can be zoomed in to understand how they are organized in the right panel. So how do you achieve this specificity? We are going to achieve this specificity by two different ways. One is either you can use antibody molecules which are specific to a particular protein or an enzyme or any structure. And I'm sure nowadays everybody knew about what is antibody. I need not describe about it, because in the COVID context, we are hearing every day about antibodies. They are naturally present in our body, which are produced by B lymphocytes through differentiated plasma cells. Since they are specific to particular epitopes, if you want to study, say, for example, actin protein in this cytoplasm of the cell, you can use anti-actin antibody to figure out where actin is present, how it is organized, and what it is doing. Similarly, tubulin you can look at with the different color. So that is how you can achieve color differentiation and locate the structure of your choice. Another alternative is if you can identify any molecule which has direct affinity towards a particular protein or a DNA molecule, you can use such dyes. So these are nothing but a fluorescence dyes or fluorochromes or chromophores, whatever you name them. They have the ability to absorb a particular wavelength of light and then emits the light in another form. So we all know the principle of fluorescence and fluorophores. And this is the most famous picture everybody must have come across. When you excite any such fluorophore, it is going to be excited to the higher state, and in higher state it is not stable, so it will come back or fall down to the ground state. During that state, it is going to liberate energy in the form of fluorescence, which can be detected by detectors. That is what is happening in case of fluorescence microscope, which is not the case with rest of the earlier microscopes, which I named up to phase contrast. Simple principle of this microscope is when you induce a light source which is passing through excitation filter, will be hitting a dichroic mirror here, and dichroic mirror has special property to reflect the light onto the specimen where your objective is kept, and then excitation of the molecule present in that objective or object will be emitting this light in the form of energy, which is transmitted through this dichroic mirror while transmitting it to the detector, that is eye, or eyes are also known as detectors. So in place of eye, you can keep a camera which can detect this. So the light which is passing through dichroic mirror, emission light, will be passing through emission filters and then reaching the detector. That is how we can detect the fluorescence. These are routinely used fluorescence microscopes. One is upright microscope. The other one is called inverted microscope. The upright microscope is so named because the objective lens are placed above the stage. Okay. Whereas in inverted, the objective lens are placed below the stage of the microscope. Specimen is always will be keeping on the surface of the stage. So if objective lens are above, it is called upright, and if it is below, it is called inverted. Otherwise, there is no difference in terms of principle. So this is what we get out of this microscope. If you are interested to look into the microtubule organization in a cell can be stained with anti-tubulin antibody, okay? And wherever tubulin is there or microtubules are there, you can see them in red emission. Same cell is also stained with centrosome specific antibody, pericentrin antibody. So pericentrin antibody is specific to centrosomes. So therefore you can see the green signal wherever they are present. You can see a cell boundary somewhere here, but here I think these centrosomes are confined to nuclear region, and you don't see anything in the cytoplasm or up to the membrane area. Third panel here, bottom in blue color, shows DAPI staining, which is specific to nucleic acids, DNA. DAPI is a DNA specific dye. You can use propydium iodide, another dye. So any color you want to choose, emission color, you can use such dyes to stain those cells. So the fourth panel over here shows the merge image of all these three. So the information of the microtubule, information of the centrosomes, and information of the nucleic acid, together we are seeing in one image as a composite or merge image, where you can clearly see centrosomes are within the nuclear region, whereas cytosol is rich in microtubule organization, and which is absent in case of nucleus. You can clearly see in this panel. So this is the power of this fluorescence microscopy. But then the drawback of this microscope is, one is resolution limit. We cannot resolve beyond this. The other one is you may get some stray signal coming out. Maybe this is a cumulative signal coming out in this single cell, which can be reduced further in case of confocal microscope. So that is how microscopy has been evolving. Still it is evolving. So what probes you have? You have several options to choose different probes, or if you can generate antibody against your choice of the protein or choice of the epitope, you can generate it and use it to probe the protein of your choice. So the some probes are listed over here for your reference. Mitochondria can be tracked by mitotracker dye, which will go and bind only mitochondria, and it stains specifically mitochondria. So as lysotracker, so as ER and Golgi, so you can essentially label different organelles using these kind of probes available. Even students can think over and generate their own probes. Okay, it's not that every time we should purchase from somebody, and they are also actually producing this based on our demand. So if students can think over, okay, to produce a dye which is economically cheaper as compared to existing dyes, you can even market them. Other probes, like in case of nucleus, we have shown in earlier picture DAPI, and you have HEXT. HEXT is actually a vital dye, and DAPI is not a vital dye. Vital dye means under live conditions you can make this HEXT enter inside the cell. In general, most of the living cells, they don't allow any chemical to enter so easily inside the cell. So DAPI cannot enter easily, but there are certain dyes which are called vital dyes. They can be entering into this cells without any fixation. Otherwise, in rest of the dyes case, we need to fix the cells. That is the biggest drawback of the this microscope. And this is a confocal microscope. It looks similar to the fluorescence microscope, but it has additional features in it. I'm not going to go to technical details because time is also not permitting. I'll quickly go through what confocal microscope is all about, and what are its main features, and how it is superior to the fluorescence microscopy. Before that, I'll just explain you the principle here. As I told you, in case of fluorescence, we are inducing incident light which is passing through excitation filter there. But here, instead of that, we are using laser light source, and lasers are very, very precise in hitting the focal plane. So laser will be passing through and then hitting the dichroic mirror the similar way as it was hitting before in fluorescence microscopy, and that is reflecting onto the specimen at different focal planes. You can see a blue arrow above and red arrow below, and middle is the yellow. So the focal plane is yellow in this case, whereas the above and below focal planes will be excluded from reaching the detector. That is the only thing that is changed in confocal microscope. In fluorescence microscope, all these above and below planes are reaching the detector, so that it is adding up to the signal, which is not true signal. Sure, you are going to see your choice of the protein, but it is amplified signal of the protein, which is actually masking the focal signal which is coming from one point. So if 2 points are there which are emitting, and their emission range is so overlapping that they are seen as single point, that is where we are losing resolution in case of fluorescence microscope, which is achieved in case of confocal microscope. So these are the important uh, functions associated with confocal microscope. One is more color possibilities, like you can go to almost 32 different colors at a time to understand a particular cell metabolism, how it is functioning and all. And less crosstalk. The crosstalk means between two fluorophores, if excitation ranges are close by, they cannot be used in this, because they are going to be emitting more or less similar kind of emission wavelength. So those two can be overlapped, and which is also known as crosstalk between the probes. So we need to select probes which are non-overlapping in their excitation range. In such case, I think you can use confocal microscope. Then optical sectioning of the specimen. This is very, very important or interesting feature brought in. We know about conventional microscopes where we generally take sections. Physically, those who come from botany background would know. Even I also did in my morphology. That means essentially you are going, or you are focusing to the signal of your interest, area of your interest, and there you are focusing uh, physically the structure of the tissue. Then once you collect the information at different depths or different focal planes, you can merge them and bring about the three-dimensional reconstruction. And this will give better resolution. For example, you can see this single paramecium, which has been stained by 7 different colors. Okay, so this is done by Alberto. This is very famous picture. You must have seen somewhere. Like these are 7 to 8 different colors, stained different structures, and captured simultaneously to know the internal details of the paramecium. That is the power of confocal. Why confocal microscopy is better? Because you can take optical sections. Okay, you can see from here left, extreme left to extreme right, you can see different panes. As you are going forward, you are seeing signal intensity increasing. Area also you can see is covering more. So that means you have to set up your start point, and you have to set up your end point, and in between these many sections you will get. So you can define through software whether you want section with a 1 micron thickness, or half a micron thickness, or maybe less than that. Depending on that, you will get different sections. So each section will have an information, and this information is put together to give rise to a three-dimensional structure. For example, this is not any antibody stained structure. It is a pollen which has autofluorescence in it, and this pollen has been subjected to optical sectioning, and the information of that optical sectioning is put together as a projection. So you can see, you can even rotate this. I don't have that movie to show you, but three-dimensionally you can rotate in 360 degrees, and you can see how this is arranged structurally in spatial resolution. Similarly, you can look at the structures. In left side, it is cells showing tight junctions in red color, red emission, and cytoskeletal structures are shown in green. And right side, it is the embryo of the zebrafish, which is stained with different fluorophores to understand their tissue organization and how that is growing further. And you can even, after reconstruction, you can rotate and animate and see how the organization of this uh, tissue of interest is. It can be an embryo, or it can be a oocyte, or any cell, anything. And everybody know or seen about mitotic spindle during mitosis process. You can even stain and take a 3D image and project it, and you can rotate and see that how these mitotic spindles are holding the chromosomes during mitosis. So such a powerful microscope it is. Finally, the resolution is another important aspect. All these features are not present in microscopy, which we have discussed before, that is fluorescence microscopy. You can just see the upper and lower panels here. This is a bacterium stained by same fluorophore. The lower half is imaged through fluorescence microscope. The upper portion is imaged through confocal microscope. So you can see clear spatial resolution achieved through confocal microscope. Okay, so that means the stray light that is giving additional or cumulative fluorescence in fluorescence imaging is reduced in confocal imaging. So that is the level of resolution you achieve, but still this is less resolution, I would say. The resolution that you achieve through cryo-electron microscopy cannot be achieved through this light microscopy. You can achieve probably few hundred nanometers resolution. Certainly it is not less than 100 nanometer resolution. So where we can use this. So far I discussed about fluorescence and confocal microscopy, but mostly we are using them on a fixed cells. But very latest technique developed by Morin et al. In somewhere in 2001, and he has actually published a PNAS paper in USA. He has developed a GFP tagging intrinsically into the genes, so that gene of your choice can endogenously express the fluorescence. I think most of you are aware of what is GFP. It is a green fluorescence protein, first isolated from jellyfish. And then people identified the protein structure, and then they have identified the genome, a genetic structure, which is coding for this. Then that codon they have used as an exon in the gene of your interest. Suppose if you are interested in actin gene to be visualized under live conditions without fixing. In live organism, you can see actin. For example, this fly is Drosophila fly, which is expressing actin protein throughout the body. Actin is ubiquitously expressed in every cell. Okay, so likewise, you can stain whatever organ of choice you are looking for. Maybe neuronal system you can, or maybe brain you can stain with the different fluorophores. Likewise, eyes of the fly here. This is ommatidial region. This ommatidial region is going to become a adult eye. Similarly, this is the testis region of the fly, where you can see single threads are nothing but one sperm, each individualized sperm. So spermatogenesis, when it is happening, this image was done. So you can clearly see, you can even count how many sperms are there in this bundle. So this technique is very, very promising and useful in understanding the intracellular dynamics of your protein of your choice, or maybe molecule of your choice. Quickly, I will explain the principle of this. The principle over here is, I think most of you are from biology background, I believe. So we have a genome which is organized into coding and non-coding regions. what we call exons and introns. So the black boxes are exons and the straight lines are introns. So GFP is constructed in such a way that it has splice acceptor, splice donor sites, and when it is targeted, it is designed in such a way that it goes and inserts into intronic regions. Maybe I can explain you this in a better way here. You have exon 1, exon 2, and exon 3, and GFP construct is targeting intron 2, and in next phase it is made an entry, and then in routine case intron 1 will be removed in this fashion, intron 2 will be removed in this fashion, whereas in this case we have additional exon added here, that is of GFP. So splicing mode will be something like this, so that you will have exon 1, exon 2, and GFP exon, and then exon 3. So GFP is brought in frame to the transcript here, and which is going to be translated and producing a protein which is intrinsically fluorescing when you observe under excitation wavelength. That is what is shown here in these pictures. These are all live images taken from organism which is normally living. Even we can label humans using this technique. Okay, there are some experiments done on that, but it is ethically wrong. Therefore, only it is now confined to animals. I would like to just show you one image captured from my laboratory related to this. Here, I think you have a Drosophila pupae. These are, it's a live one, and this is endogenously labeled by such GFP molecule into the eyes. Okay, this is the anterior position. What you are seeing, a thick to big GFP signals. Each one is one eye. So eye has been targeted here for developing a cancer. Like one of my research student is working on cancer, so he has used this technique in this particular aspect. So we would like to see how this organism is dying. We know that at particular point in time it is dying. How it is dying, we would like to capture. The right side of the image, it is already dead one, and you can see these two spots are totally distorted and organism is dying. So we would like to understand the phenomenon, how this is happening under live condition. So you just concentrate on these two big dots, two eyes, which are nothing but eyes which are connected to the brain region. These optic lobes are actually going to be positioned to laterally. You see how beautifully they are positioned to laterally. At the same time, brain is expanding at that region. So this entire region is actually brain region. Hours of hard work, this image. You can just concentrate on that, how beautifully it is laterally. Still it is living, okay? And this is a normal wild type fly harboring GFP without any problem. It is living, and which is actually distorted in case of cancer. I will show you the cancer movie, how that is going to be dying. To make it faster, I will go through. See, this is a point where it is died. So due to cancer, there is a cell-to-cell contact is affected, and as a result, the positioning, lateral positioning is affected, and the organism is dying there. So we can concentrate on this area. What are the events that are going to go wrong there? how can we prevent that so that we can prevent the cancer. So this way we can use effectively the live imaging to understand a biological question, to address a biological question. Next, coming to super resolution microscope. This also looks like confocal microscope, and it is confocal with additional feature of emission depletion, which is resulting in additional resolution. The way confocal is getting better resolution because of the cut off the light through pinhole. Here also there is a technology developed by a Leica company that is called STAD, and Zeiss company is also working on this in SIM technology, structure illumination technology. So through this you can get further information within intracellular compartments. Like you know that a cell has nucleus, a nucleus has nuclear pores. You can see a nuclear pore organization through this microscope. That much resolution it gives, but beyond this it cannot give. You cannot see individual supramolecular structures of this nuclear pore complex, which is actually schematically represented here. This structure you can better resolve in cryo-electron microscopy. So I'm not going to go through all this due to time constraints. Like you can target any cytoskeletal components, you can target endoplasmic reticulum, mitochondria, different organelles, and understand the structural aspects of them. Just to show you the difference between a confocal microscope and super resolution, I just made this slide. A confocal microscope gives you a picture like this, something like this. You have information, but you don't have that spatial resolution, which is seen in case of super resolution. You have a nucleus which is stained in blue, and you have actin filaments stained in, say, green, and you have mitochondria stained in red. So you can clearly see distinct mitochondrial dots. Another thing I would like you all to remember, this dot-like structures mitochondria, we can only resolve up to here in this light microscopy. I'll be showing the same thing in cryo-electron microscopy, to what extent information you are going to get. Okay? You just remember this picture in your mind. In the other right side panel, you can clearly visualize the resolution difference from confocal versus super resolution. Similarly, bottom panels in extreme left is showing nucleus in blue, mitochondrial organization in green. Okay? You can see very big mitochondria, which are actually looking like threads. So mitochondria, interestingly, can be seen as circular as well as thread as well as complex networks. But routinely in textbook, we see only mitochondria, which is registered in our mind. But in reality, we have a lot of mitochondrial networking. Sometimes we can see them as tubes, big tubes. Okay, so that is another interesting area to be worked on. I'm working on that area. So I would like to show you a 3D animation taken out of this. If I can show you this. So this is the cell which is dividing. Okay, so you can clearly see mitotic spindles along with the chromosomes attached to it and other cytoskeletal structures. So this you can achieve through super resolution microscope, which cannot be seen through fluorescence. You can see through, but you cannot get this resolution. So we can take images captured and then reconstructed in the form of, okay. Now I'm getting into a cryo-electron microscopy, which is very, very revolutionized microscopy.
And inside the column, there's very high vacuum.
Sorry for interruption. So these three scientists really worked hard for past 30 years to develop cryo-electron microscopy, for which they received Nobel Prize in 2017. Uh, Henderson is in UK MRI, UK lab, Cambridge University, and he is the man behind this entire imaging process. And the Joachim Frank from USA, he developed a mathematical algorithm, and the Deboche, he has developed vitrified ice formation. So all these three together, clubbed together, we got the cryo-electron microscopy as the end product. So they got this honor for developing this technique, which enables unprecedented views of biomolecules. Okay, for example, this is a protein molecule. In the left side, what you see is a protein which is having less resolution. You cannot see, okay, beta sheets or alpha helices or even side chains of the amino acids. So this is less resolved structure, which is taken by, okay, conventional microscope, whereas the cryo-electron microscope resulted the structure which can be seen very clearly, even the R chains of the amino acids. So that is possible without crystallization. That is the key feature of this microscope. We all know Watson and Crick discovery of DNA double helix. They have used a conventional crystallography technique, and through X-ray diffraction, they could identify the structure of the DNA double helix. But for conventional crystallography, we need to crystallize the molecule of our choice first. So without crystallization, you cannot achieve structure. That is where these scientists looked for an alternative, because every molecule cannot be crystallized. Size matters, or many parameters matters. So with very great difficulty, you may crystallize something. Maybe you cannot achieve the highest possible resolution even after crystallization. So to overcome that, they have created another alternative technology through which they can be forming a vitrified ice in which these proteins must be embedded, and that vitrified ice can be subjected to imaging through cryo-EM, and then processing those images that they get through 2D classification and 3D classification. Finally, the final structure can be achieved. So that is where they got the Nobel Prize. How it looks, this microscope is very gigantic. It is almost double the size of myself. This is at Stanford, where I worked with this system. This is the latest microscope available in the world, which is known as Krios microscope of 300 kilovolts energy. So if you see internal anatomy of this microscope, it is so complex. You cannot really understand the hardware of this microscope. But it gives excellent resolution and information. In the present context, when we are facing the COVID pandemic, everybody is talking about spike protein. But how do you understand the structure of spike protein? You can understand that only through this means. Or you can even understand through crystallography. But as I told you, crystallography has its own limitations. You cannot crystallize all the proteins. And the other thing is time factor. Now in pandemic, we need everything in very fast mode. The cryo-EM can give you a structure resolution within a week time. Whereas in crystallography, to crystallize a protein of your choice would take anywhere between 2 years to 10 years. Even after crystallization, there is no guarantee that you can get the structure of your choice. So that is where this is the only alternative available to understand the structure. But so far, only limited information is worked out in case of COVID-19. I will talk about that a little bit later. The extreme right has the principle of this microscope. You have electron beam coming from the top, okay, from the electron gun existing on the top of this somewhere here, and it is pumping through a vacuum, and it is passing through vacuum. While it is passing, it is hitting the object. it is passing through objective lens and hitting the object which is present on the grid of the specimen. These vitrified ice are formed on the grid surface of the gold grids or maybe carbon grids, where the proteins are embedded in vitrified ice. Vitrified ice is not a crystalline ice. Vitrified ice is something which is very flash frozen, thing. Normally, when you keep water into a deep freezer, it will be crystalline in few minutes to hours. So that is called the crystalline ice, but that is extremely dangerous for this microscope. You cannot get anything out of crystalline ice. That is like considered to be a contaminant of the microscope. So what we have to achieve is a vitrified ice, which is not like a crystalline, but when it is frozen in a quick manner in minus 195 degrees Celsius temperature in the liquid nitrogen, it is going to be crystallized at random. The molecules which are present in the that ice area are randomly organized, therefore it is called vitrified ice. So within that randomness, your protein of choice will be embedded in a different orientations. I can show you a movie where you can understand how that is achieved. A little later I will show you. So once this is beam is passing through this, it can capture two-dimensional image. There are three proteins shown here, and you can get three proteins detected by a direct detection system. This is nothing but a detector of the cryo-electron microscope. This is direct electron detector. The camera, which is very costly. I think one camera may cost around 6 crores to 9 crores rupees in Indian rupees. That camera has the power to capture electrons directly. So these captured images in two-dimensional form can be categorized, classified into different orientations. Where we can categorize them into different classes. So all those molecules, suppose there are 1 lakh protein molecules you have captured in your image, maybe 20,000 molecules are of one orientation, and 10,000 are of another orientation, 25,000 are of third orientation. Likewise, you should be able to get as many as orientations possible to solve the structure. That is the tricky point of this microscopy. So once you get all these classes, then you can flow through a workflow to get a three-dimensional composite image, and which is the final image of the structure that you are looking for. If it is COVID-19 spike protein, you have to subject spike protein through this process to get the final spike protein image. We all know that, especially after the COVID pandemic, we know that how virus can attack our body and what changes it can bring about, and how cells are getting affected, how immune system is acting against. Everything is known to almost even general public. So we know that our body is made out of complex tissues, which are made out of cells, and each cell has its own metabolism and it has its proteome, and we need to look into each protein of that cell relevant to the problem that we are looking into. Uh, COVID is a medical problem or societal problem. So if you want to know what are the surface proteins that are interacting with the spike proteins, then you can perhaps design a better diagnostic kit, or you can design a better therapeutic target, or you can design a better vaccine. So for that purpose, we need to understand the proteins first, surface proteins especially, which are there in the membrane of the cells, which are interacting with the spike protein. And we all know that genes are responsible for protein production, whether it is virus, bacteria, or human being. So every organism has its own genome, and it is coding corresponding proteins, and these proteins are actually getting inside, interacting with each other, and communicating with each other by chemical means, and internalization process is happening. A virus always looks for its own reproduction or amplification inside the host, and host always try to block its reproduction or infectivity by means of immune system. So I think the lower panel shows how can we understand the protein structures through cryo-electron microscopy. I think which I have already explained you. You can take protein of your choice in the tube, subjected to grid preparation, sample preparation, and then go for imaging. Here, while doing sample preparation, I can show you maybe a video if my internet is on. Yes. Uh, so this is called vitrobot. We are going to fill this container with liquid nitrogen, because everything that has to be processed must be within minus 185 to 195 degrees Celsius. Therefore, this microscope is known as cryo-electron microscope. We need to set parameters like humidity, temperature, and other conditions, blotting conditions. For example, here 174 is the minus 174 is the temperature. Then we need to fill liquid ethane into a container which has slightly higher temperature than the liquid nitrogen. And we need to insert a blotting paper here and mount the grid holder. This is the grid, very thin grid of say 2 to 3 millimeter size. So we are going to apply sample on the one side of the grid. For example, you can take spike protein of the COVID-19 and you can apply onto the grid to solve its structure. You can set up blotting time for say 2 seconds, sometimes 3 seconds. So now the protein sample is being applied onto the grid here. So it is applied onto the grid. Then blotting is initiated. Excess fluid will be absorbed by blotting paper. It immediately plunge freezed into the liquid nitrogen. This is called plunge freezing sample preparation technique. So now sample is rapidly frozen into liquid ethane, which is maintained in liquid nitrogen temperature. And henceforth, the sample has to be in liquid nitrogen temperature, so minus 185, till you image. So after that, I think you can, after image, you can see the protein structure, something like this. This bright area is called whole area, which is thinnest possible area on the grid. And this darker area is actually grid area. If any sample is falling onto the grid area, this cannot be imaged. This is very thick to be imaged. We can only image the protein molecules which are falling onto this hole. So each grid will have squares, around 1000 squares, In each square, you have few hundred holes. Each hole is of say 2 nanometers thick. So we can only see, sorry, 2 microns thick. We can only see the protein or maybe cells which are part on this whole area. So we can pick up each protein as many as if you can pick up, the better would be your structural uh, resolution. So these kind of structures you can identify through that protein. Okay. Once you image and classify them and finally resolve the structure, you can get a structural resolution as as high as 1.9 angstroms. This protein is, everybody knows it is beta galactosidase from E. coli, and even school kids also knew about beta galactosidase. Extreme left is less resolution image, extreme right is high resolution image, and you can clearly see the beta sheets and alpha helices, and you can see the symmetry of the enzyme, and perhaps this is the active site region of the enzyme. So once you get the structural information, you can go ahead and understand its substrate binding or maybe inhibitor binding sort of things. So since time is limiting, I will quickly go through this. Uh, Coming to the corona, so everybody wanted to know about, and we are on webinar because of this. In light microscopy, corona looks like this. I have already shown you. Each spot, yellow spot that you see in light microscope of scanning electron microscope, it is not even light microscope. I'm sorry, this is a scanning electron microscope image. In light microscope, you cannot even see this dot. Okay, but you can maybe achieve that by antibody staining somehow. But this is scanning electron micrograph. And this small dot, when you look into the transmission microscope, you will see this kind of resolved image.
Professor C. Krishna, sir?
Yeah.
Around
Yeah.
how much time would you complete, sir?
Yeah, I will be finishing in 5 minutes.
Yes. Thank you, sir. Thank you.
Thank you. So then quickly I can just tell you about the spike proteins which are present on this virus can be resolved to this extent. As I told you in the beginning, in the previous picture, this is the resolved surface protein structure of this spike protein. Further, that spike protein can be glycosylated. The pink areas are glycosylated areas, yellow areas are glycosylated areas. That can also be understood by cryo-electron microscopy. These are different viruses. Just to have an idea, I kept them into one picture. Most of the viruses, they have their surface proteins which are functioning in similar manner. As I told you, these surface proteins can be resolved up to the backbone of the peptides. One just tomography I can show you and then finish. For example, this is the mitochondria which I took from the same machine. You can see a mitochondria with double layer membrane, and cristae, they are seen inside the mitochondrial membrane. So in tomography, we take -60 to +60 different frames. These are gold fiducials as a reference. -60 has given those subframes clubbed together, and now +60 will be giving the rest of the information. So in this, you can clearly see the cristae resolved, which cannot be seen through light microscope. Maybe I will stop this here. And this is the cancerous mitochondria, where you can see a double membrane here, but you have a lot of very electron dense thick aggregates present in this cancerous mitochondria along the cristae. So that is interesting feature which we are working on in collaboration with Stanford. Okay, so this information you cannot get through a light microscopy, nor through conventional crystallography, because these are tomograms. And then the final slide I would like to show you is a correlative cryo-electron microscopy. We have seen light microscopy of various types, and then we have seen cryo-EM. Clubbing light microscopy with cryo-EM is nothing but correlative cryo-EM. I'm not going to go into details. What we can understand from this is, first you label whatever molecule of your choice in a big cell. Cells are cultured on the grid directly, subjected to plunge freezing, and then imaging is done first through a confocal cryo microscope. This is called a confocal cryo microscope. Everything is done at minus 185 temperature. And then grids can be subjected to imaging. One square can be picked up, and in that square you can see the choice of your molecule, and that can be marked, and then you can go for imaging of that to get the structural resolution what you are looking for. So that is what is very latest technology which is there in the world, maybe in 1 or 2 labs. Not every lab has this technology. I'm trying to get this technology to India. Let's hope so, because it's very costly affair. The technology involves scanning electron microscope beam. It is called fib milling, and we have to make very thin layer of structures within that vitrified ice to achieve these structures. Okay, I have a movie to show that, but since time is not permitting, I can skip that. I would like to thank this Grant. He is my role model, and I suggest every audience to go through this website and register yourself. Okay, I can provide you later also. It is emlearning.com, where you can get entire information about the cryo-electron microscopy, and his lectures are very, very interesting. I wish you all the best, and hope I could convey you some message out of this webinar. Thank you very much. Hello. Yeah, thank you very much. I think I finished.
Yes, sir. Thank you very much.
Now it is open to questions. If any
Yes,
audience.
yes. We will ask. Maushmi is moderating. What happened? There is some this thing. So we probably will have time just for 2-3 questions, sir. We'll address the questions to you later in your mail, sir, so that they can seek an answer. Is Maushmi coming in to ask questions quickly? There is
Ma'am,
yes.
yes, ma'am. This is Dr. Anupama. I'll take over this. So here the first question is, sir, what is cutting edge benefit of cryogenic electron microscopy over nuclear magnetic resonance spectroscopy in structural studies of virus?
Okay. Nuclear magnetic resonance NMR can give you structures which are very, very limited in nature, whereas cryo-electron technology through this microscopy can give you structures as big as even cells. Okay, in NMR we can perhaps limited to very, very narrow range of structural elucidation. For example, the spike protein is a big protein. Okay, so you cannot really work out spike protein structure through NMR, but a portion of that could be worked out through NMR. Whereas in cryo, you can work out the entire protein. Okay, especially the bigger proteins can be easily worked out through cryo-EM. One limitation with cryo-EM is you may not be able to achieve a good resolution below 100 kilodalton proteins. So far that is the limitation. Although some groups clearly showed up to 17 kilodalton. Proteins are also solved through this microscopy, but it is a bit difficult to solve. Okay, so that is what I understand from this question.
The second question is, though cryogenic electron microscopy principle is more than 4 decades old, isn't it took long for evolution process to have significant impact in bioimaging until recently?
Yes, cryogenic technology is not new, it is old. The way transmission microscopes are used way back in 1960s, okay? But cryogenic microscopes are also used since maybe mid 70s and also. But the revolution these three scientists brought into this cryogenic technology is plunge freezing vitrified sample preparation, which was not there before. This has achieved only in this 2016 to before that, for which they got the Nobel Prize. And this vitrified thin layer of ice formation made us to understand the structural features of the protein. Before that, we have no way to solve the structure except conventional crystallography. That means, uh, for example, if you want to study the COVID-19 spike protein, you can only crystallize the spike protein. Once you get crystal, then only you can get the information. But the kind of unprecedented, uh, less than 2 angstrom resolution is not possible otherwise before. Therefore, it is cutting edge technology.
Thank you so much, sir. As we are out of time, almost we are reaching the end of the session. So I
Okay.
thank Professor Sri Krishna for his valuable time. And I hope all the audience, they are enlightened with this microscopic techniques. Thank you, sir. Thank you once again.
Thank you very much for your time. And
Thank you, sir.
thank you. Thank you.
Thank you. So, dear participants, actually, we are supposed to have a second session on the trends of biomedical engineering, which I was supposed to give you the introduction to. But since we are lagging time, I'll just tell that biomedical instrumentation, it has penetrated into our houses. It is there everywhere. Please look around yourself. We have lots of equipment which we are using, like the chairs, the wheelchairs. Like great people have been using this. Like we have this example of Mayuri Sudhagaru, who has used these devices, the prosthetic leg, and then you have the Blade Runner who is using it, and more advanced things are coming up in this biomedical engineering. But the need of the day is all of us have to contribute to this field. This is a highly interdisciplinary field. Let us contribute to the field and do our best part to fight COVID. We have Innovation Hub opened up by WHO where you can deposit your thoughts. Thank you for that. And as we are running out of time, let me propose vote of thanks. As a coordinator, I deem it a privilege to propose the vote of thanks and thank everyone who has helped in making this webinar happen. Firstly, I thank Professor S. Sri Krishna from Banaras Hindu University. In spite of your very busy schedule, you accepted our invitation and took up the session on a very challenging area, and being passionate about your subject is evident in your presentation. Thank you so much, sir. I thank our principal, Dr. Sister Shaiji, who stands by us and makes these webinars happen. I thank our vice principal, Sister Hema, and secretary, Sister Mary, who always extended their unconditional support. I am grateful to Professor P. Sharada, IQAC coordinator, who guides all the departments, including ours, in planning webinars. It is time to thank Ms. Ramni Dhanraj ma'am, who is a member of IQAC and is the head of the Department of Office Management. Her presence adds value to every event, and she stands as a pillar for us. I have no words for the technical help rendered by Dr. Gauri Shankar sir, Mr. Srinivas sir, Chandni ma'am, and everyone there who have been working tirelessly to make sure the event runs smoothly. I thank my colleague, Dr. Mausmi Shankar, who is ever ready to support me in all events planned from the department. I thank each and every member of the administrative and technical team involved in organizing the webinar. And finally, I thank all you participants whose overwhelming response for the earlier quiz which we have conducted and this webinar makes us feel short of words. Thank you so much. Thank you everyone for your responses. And please fill up the feedback form now, and you'll get your e-certificate shortly. And let us all stand by for national anthem.
YouTube logging, not the info. So, we got a feedback.
So that was again a very good webinar by the Biochemistry Department, organized and coordinated by the Biochemistry faculty, Dr. Mary Anupama and Dr. Maushmi Shankar. Let's meet again for another interesting webinar from the platform of St. Joseph's College for Women. A big round of applause to the Biochemistry Department. And please be with us, participants, for a lot more webinars. We'll see you often, and we believe in quality and excellence. Jai Hind, good day, have a nice day. Thank you.